Hello everyone, how are you all? I hope each and every one of you is in the pink of health and happiness. So shall we start the class? Am I audible, visible? Yes. So today we are going to learn about molecular basis of inheritance. One of the highest weightage chapter, most important chapter, entire syllabus, or out of entire syllabus is the most important chapter, that is molecular basis of inheritance. So as usual we are going through the PYQs one by one. Along with that we are going to learn the concept. Okay. So shall we start? Our first question is, match the list. Yes, match the list 1 and 2. Histones. Histones are
Positive.
yes. Histones are positively charged basic proteins. It's on. Yes, it's on. It's on. Yeah, it's live. So, nucleosome. Nucleosome is
DNA wrapped around
DNA wrapped around the histone octamer. Yes. Euchromatin is loosely packed. Heterochromatin is densely packed chromatin. Okay. 1 A3, A3, B4. We got D4 Delhi as a correct answer. Understanding? Yes. Given below are two statements. Statement 1, in prokaryotes, the positively charged DNA is held with some negatively charged proteins in the region called nucleoid. Is it correct?
No.
No. And in eukaryotes, the negatively charged DNA is wrapped around the positively charged histone octamer to form nucleosome.
True.
True. So it is, one is incorrect. Statement 2 is true. So option A is the correct answer. Very good. Now match the list 1 and 2. Haploid content of human DNA is 3.3 into 10 to the power 9 base pairs. Escherichia coli,
4.6.
yes, 4.6 into 10 to the power 6 base pairs. And bacteriophage lambda is 48502 base pairs. Bacteriophage, this one, pi 5174, 5386. So A2, B1, C4, D3. D4 Delhi is the correct answer. If the length of DNA molecule is 1.1 meters, what will be the approximate number of base pairs? In a diploid content, it is 6 points.
6.6.
Oh, okay, okay. This is okay. It should be power. Okay. Yes. A slight mistake. Okay. Yes, it is. If it is 1.1.
3.3 into 10 to the power 9.
3.3 to 10 to the power 9 base pairs. Yes, this is the haploid content of DNA, isn't it? The DNA molecule is around 2.2 meters normally, diploid cell, but if it is 1.1 meters, it is 3.3, that is half of, that is haploid content, you can see. Yes. Read the following statement and choose the set of correct statements. Euchromatin is loosely packed. Is it correct?
Yes.
Yes. Heterochromatin is transcriptionally active.
No.
No, it is inactive. Yes. And euchromatin is transcriptionally active. Yes. And histone octamer is wrapped by negatively charged DNA in nucleosome.
Yes.
Yes. histones are rich in lysine and arginine.
Yes.
A typical nucleosome contains 400 base pairs of DNA helix. Actually it is, it is no. Actually it is 200 base pair. Typical nucleosome contains 200 base pair of DNA helix, which means if this is a octomer, this wraps around 2 twice and go, and if this is a octomer, then again wraps around like this. So this wrap is 146 base pair wrapping and 54 base pair here, the hanging DNA here. So totally from here to here, if it is one nucleosome and the H1 is here, then it will be 200 base pairs, not 400 base pairs. So this is wrong. We need to find the correct answer. So it is A, C and E. Sorry, A, C and D. C is the correct answer. Any doubt? Complete the flow chart. This one A is replication, B is transcription, C is translation, okay, and D is protein. Eliminate it, eliminate it. Replication, transcription, translation and protein. Okay, so it is B is the correct answer. If adenine makes 30% of the DNA molecules, what will be the percentage of thymine, guanine and cytosine? Tell me. The content of adenine is equal to the content of thymine. So here they have given it as 30%, which means thymine is also 30%. If this is 60% total, this will be
Option B.
Yes, very good. This will be another 40%, which means 100 minus 60. You know it, basic math, yes. So if it is 40, then it should be 20, 20. So TGC 30, 20, 20. 30, 20, 20 कहाँ है? Yes, yes. What did you say? Option C. What, Sakshi?
Option B.
Yeah, option B. Yes, option B. Because T is 30. Directly you can say from A, you can say 30. We don't have to even do that, because here directly we would have eliminated the other options, isn't it? So we would have directly got B as a correct answer. But this is how we do this. Which one of the following statement about histones is wrong? The pH of histone is slightly acidic. Histones are basic.
Basic.
Yes, they are basic. So we need to find the wrong one. We got it. A mein we got it. The wrong option. Histones are rich in amino acid lysine and arginine. Yes, basic amino acids. Histones carry positive charge in the side chain. Yes, they do. Histone are organized to form the unit of 8 molecules. Yes, octomer. Histone octomer 8. Which of the following statement is correct? Adenine pairs with thymine through 1 hydrogen bond? No. 3 hydrogen bond? No. It is guanine.
2.
Yes, this guanine and cytosine does with a 3 hydrogen bond. Does not pair with? No. It is 2 hydrogen bond. D for Delhi is the correct answer. If the distance between two consecutive base pair is 0.34 nanometer and the total number of base pairs of the DNA double helix in a typical mammalian cell is 6.6 into 10 to the power 9, okay, 10 to the power 9 base pairs, then the length of the DNA is approximately, direct NCERT question, directly NCERT में लिखा हुआ है, it is
2.2 meters.
2.2 meters directly. Otherwise, if you multiply that, also you will get 2.2 meters. In a polynucleotide chain of DNA, a nitrogenous base is linked to OH of sugar में which prime? Nitrogen base. ये होता है. This is 5th carbon. This is 4th carbon. The different. Okay, this is 5th carbon. 4th, 3rd, 2nd, 1st. यहाँ पूछा गया है नाइट्रोजन बेस linked to the OH of first carbon. Any doubts? Not 3rd carbon. Yes. So it is C is the correct answer. 1 prime carbon of the pentose sugar. E. coli has 4.6 into the 6 into 10 to the power 6 base pairs and completes the process of replication within 18 minutes, then the average rate of polymerization is approximately. This is directly concept based question. Tell me.
Option D.
200 base pairs per second. 2000 base pairs per second. Option D is the correct answer. Understanding? Directly you can say 2000 base pair per second. Purines found both in DNA and RNA are purines. Thymine is a pyrimidine. Purines are adenine and guanosine. The association of histone H1 with the nucleosome indicates transcription is occurring, DNA replication is occurring, the DNA is condensed to a chromatin fiber, DNA double helix is exposed. Association of H1 with the nucleosome indicates?
Option C.
Option C, very good, this is the correct answer. The DNA is condensed to form chromatin fiber, which means like DNA is there, then it is wrapping around the octomer and this H1 is sealing this one, sealing the wrapping. This is the H1. Understanding? This is H1. Okay, H1 histone. And here, इसमें octomer है. This one. This has the octomer. Understanding? So, which means the DNA is condensed to form the chromatin like this. And this is sealing it. This is sealing like this. So, it is condensed to form the octomer. The DNA fragments are positively charged, negatively charged, neutral, either positively or negatively charged depending on their size. Easy?
Negatively charged.
DNA fragments are negatively charged. B for Bombay is the correct answer. Identify the correct order of organization of genetic material from largest to smallest. The largest is, of course, genome. So, you can eliminate these two. Then comes the chromosome. Then comes the nucleotide. Nucleotide is just one, right? So, then comes the gene, then comes the nucleotide. It is genome, chromosome, gene and nucleotide. We need to find the largest to the smallest. Yes, B for Bombay is the correct answer. Any doubts? The diagram shows an important concept in the genetic replication. Genetic implication of DNA. Yes, fill in the blanks. So, A is a transcription, translation proposed by Central dogma of inheritance, Watson and Crick. So, transcription.
Option D.
Yes. Option D. Transcription, translation, Francis Crick. Yes. Unequivocal proof that DNA is the genetic material was first proposed by who?
Hershey and Chase.
Hershey and Chase experiment. Yes. It was a unequivocal proof. Hershey and Chase experiment. Yes. Given below are the two statements. RNA mutates at the faster rate. Is it true?
Yes.
Yes, it is unstable and mutates at the fast rate. So, statement 2, virus having RNA genome and shorter lifespan mutate and evolve faster. Is it true? See, as the RNA can mutate faster, the virus having RNA genome can also mutate faster. If this is true, then that is also true, right? Viruses having RNA genome and shorter lifespan mutate faster, because RNA as a genetic material is quite unstable compared to DNA. So many different, that's the way viruses we find so many different strains. For example, common cold ka virus lo, how many different strain we have. That's why we get, even though we get the immunity if we once infected with that particular virus, but they have thousands of strains. So, we will be infected with different strains. That's why the common cold is like so common, isn't it? So, both are true. Both statement 1 are true. B for Bombay is the correct answer. Which scientist conducted an experiment with this one, radioactive phosphorus and sulfur, labeled for demonstrating DNA?
Option B.
Yes, Hershey and Chase experiment. The unequivocal proof for giving, for saying DNA as a genetic material. Statement 1, RNA being unstable. Yes, RNA being unstable mutate at the faster rate.
Yes.
Yes. RNA can directly code for synthesis of proteins, hence can easily express the character.
Yes.
So, both 1 and 2 are correct. C is the correct answer. With reference to Hershey and Chase experiment, yes, select. Virus grown in the presence of radioactive phosphorus contain radioactive DNA.
Yes.
Yes. Virus grown on radioactive sulfur contain radioactive protein.
Yes.
Yes. Virus grown on radioactive phosphorus contain radioactive protein. Virus grown on radioactive sulfur contain radioactive DNA. No. The virus grown on radioactive protein contain radioactive DNA. So, A and B are the correct answer. We need to find the most appropriate option, which is A and B. B for Bombay is the correct answer. If DNA contains sulfur instead of phosphorus, and protein contains phosphorus instead of sulfur, what would have been the outcome of Hershey and Chase experiment? Tell me. Here there is a condition, if the DNA contains sulfur instead of phosphorus, DNA has phosphorus but they are telling it contains sulfur. What if it contains sulfur and the protein contains phosphorus not sulfur, then what would be the outcome? So, if the bacteriophage, bacteria, and they are saying the DNA contains sulfur. So, this is the radioactive phosphorus. They are telling the protein contains phosphorus and this is the DNA contain sulfur. So, when it inject inside, the bacteria has now the genetic material of the virus. So this is sulfur here. According to this condition, this is sulfur here. Understand? Now options. Radioactive phosphorus in the bacterial cell? Nahi. Radioactive what? Sulfur aa gaya hai. So this is wrong. No radioactive sulfur in the bacterial cell? Sulfur to aa gaya hai na? Nahi. Wrong. Both radioactive sulfur and phosphorus in the bacterial cell? No. The outer coat will go out. Only the genetic material will get into the bacterial cell. Both radioactive sulfur? नहीं हो सकता. Radioactive sulfur in the bacterial cell? ये देखो sulfur आ गया ना, radioactive sulfur. According to the question, they said DNA contains sulfur, which means the radioactive sulfur is in the DNA. It has come into the bacterial cell. So D4 Delhi is the correct answer. Tell me yes or no? Yes or no? Yeah. Read the question very carefully. Okay? We could able to solve it if we read the question carefully. Yes. So D4 Delhi is the correct answer. Radioactive sulfur in the bacterial cell, because according to the question, the sulfur has come into the bacterial cell, because these bacteriophage were growing in that particular radioactive sulfur. If the DNA has the sulfur, the DNA got this radioactive sulfur into that. 10 E. coli with 15 the isotope of nitrogen, which is a heavy isotope, heavy isotope 15. Okay. dsDNA are incubated in the medium containing the normal this thing of the nitrogen 14 nucleotide. After 60 minutes, how many E. coli cells will have DNA totally free from the heavy isotope of nitrogen? Tell me. It is, they are telling 10 E. coli cells were there which were grown in heavy isotope nitrogen. Okay. Now we are starting from here. E. coli can divide every 20 minutes. So one cycle, next 20 minutes, another cycle, next 20 minutes, another cycle. At the end में कितना है पता लगाते हैं. So they are telling after 60 minutes 20 plus 20 plus 20 to 60. So in first 20 minutes there will be double 10 20 cells. So in them, according to semi-conservative model, so in half of them there will be half of the DNA content here will be having heavy isotope of 15, nitrogen 15, and half here will be having heavy isotope of 15 and nitrogen 15. So next 20 minutes this will become 40, isn't it? In that 40, the 20 here will be containing this nucleotide, the DNA with the nitrogen of heavy isotope. Next will be containing the normal ones. Oh, not this. So on. So next 40 will become 80 here. In 80 also 20 will be containing this, another 60 will be containing normal. We need to find how many E. coli cells will have DNA totally free from. Tell me how many? How many? Okay. So for example, 1 will not take 10, 20, 30, 40, okay fine. For example, there was one bacteria. In that one bacteria, it was having both. It was grown in heavy isotope 15, nitrogen. So it was having this particular DNA. After 20 minutes, it became 2. After 20 minutes, the bacteria divided. The bacteria divided and it became 2. According to semi-conservative model, what happens in both the bacteria, one strand will be heavy isotope and the another strand will be normal isotope, isn't it? Normal one, isn't it? So again this will become, after 20 minutes ये खुद divide होके 2 हो जाता है. In that what happens, ये स्ट्रैंड का और एक स्ट्रैंड बन जाता है, ये स्ट्रैंड का रेड का और एक स्ट्रैंड बन जाता है. ओके ओके. और इस वाइट स्ट्रैंड का क्या होता है, दिस वाइट स्ट्रैंड का और एक स्ट्रैंड प्रिपेयर होता है. सो आफ्टर 20 मिनट्स यहाँ 2 थे ना, यहाँ 2 थे ना, देन अगेन डिवाइड एंड बिकम अनदर 2, राइट. सो यहाँ दिस बिकेम 4, 1 2 3 4, अंडरस्टैंडिंग. एंड इन दिस ऑल्सो फ्रॉम 4 it become in 20 minutes it will double it, so 4 became 8. in 8 इसमें देखो ये दोनों स्ट्रैंड यहाँ हैं, अंडरस्टैंडिंग. फ्रॉम दिस वाइट स्ट्रैंड another normal isotope, that is red, will become, and from this white, one white is there, another red, understanding. and यहाँ में 1 2 3 4 5 6 इनका 1 2 3 4 5 6 normal isotope 14 will become will come as a semi conservative model में, understanding. So we need to find how many E. coli cells will have DNA total free from 15 nitrogen, that is heavy isotope 15 of nitrogen. How many? So in that we did for 1, right, just multiply into 10, it will be 6 into 10 60, which means 60 will be having the DNA totally free from the heavy isotope of nitrogen. Not understood? Not understood? Okay, we'll do again. शुरू से शुरू करेंगे, शुरू से. Yes. Am I audible, visible? First, is there any? Yes. So there were, shall I take one as an example first? Shall I take one example? We'll divide into 10. Sorry, we'll multiply into 10. Multiplication, division of cells, multiplication of cells. Yes. So this is the beginning. Okay, we have one cell which was grown in heavy isotope 15, okay, of nitrogen, heavy isotope 15 of nitrogen. एक सेल था वो ग्रो हो रहा था हेवी आइसोटोप में, ठीक है. तो हमें पता लगाना है कि 60 मिनट्स के बाद क्या होता है उसके साथ. इट वॉज इंक्यूबेटेड इन हेवी आइसोटोप्स, बट हियर यहाँ 60 मिनट्स में वो नॉर्मल आइसोटोप दैट इज 14 में इंक्यूबेट हो रहा है. ओके, दिस इंक्यूबेटेड इन द मीडियम ऑफ नॉर्मल, जो दिस वन नाइट्रोजन का नॉर्मल एटॉमिक वेट होता है ना, दैट इज 14, उसमें इंक्यूबेट हो रहा है, नॉट द हेवी वन. ओके, उसका कोई आइसोटोप में नहीं, नॉर्मल वन. सो इट इज इंक्यूबेटिंग नॉर्मल. वी नीड टू फाइंड आफ्टर 60 मिनट्स में क्या होता है. चलो 60 मिनट्स मतलब इन वन साइकिल ऑफ सेल डिविजन टेक्स 20 मिनट्स फॉर ई कॉलाई. सो 60 मिनट्स में 3 साइकिल हो जाएंगे. दैट्स वाई वी आर डिवाइडिंग इनटू 1 2 एंड 3. ओके, 1 से 2 होने के लिए 20 मिनट्स चाहिए, 1 से 2 होने के लिए. ओके, एंड 2 से 4 होने के लिए another 20 मिनट्स, 4 से 8 होने के लिए another 20 मिनट्स, another 20 मिनट्स. सो टोटल मिनट्स कितने हुए? टोटल हो गए 60 मिनट्स. तो वो भी वही पूछा गया है क्वेश्चन में, 60 मिनट्स के बाद क्या होता है. तो एक बैक्टीरिया है ये, उसके अंदर हेवी आइसोटोप, दैट इज both the strands of DNA are heavy isotope, 15 ही है heavy isotope of nitrogen. Okay, when it divides तो क्या होता है? When it divides according to the semi conservative mode of replication, इसमें एक स्ट्रैंड इधर चला जाता है, एक स्ट्रैंड दूसरे में चला जाता है ना, एक स्ट्रैंड इधर चला जाता है, एक स्ट्रैंड दूसरे में चला जाता है. सो अनदर स्ट्रैंड कौन सा हुआ? मैं वाइट से इंडिकेट कर रही हूँ, ये विच इज ये स्ट्रैंड है ना, दिस स्ट्रैंड, दिस इज नॉर्मल नाइट्रोजन का हेवी आइसोटोप नहीं है, इट इज़ अ नॉर्मल एटॉमिक वेट 14 ही है. ओके, मास, एटॉमिक मास 14, नॉर्मल एटॉमिक मास. ओके, सो नाउ इफ 1 became 2, मतलब इधर इसको इधर डाल, सेम इधर आई एम मल्टीप्लाइंग, आई एम राइटिंग इट हियर. सो 1 was this strand, like नॉर्मल, ये तो N14 का स्ट्रैंड है, N14. ओके, एंड द प्रीवियस स्ट्रैंड एज सेमी कंजर्वेटिव में दिस इज द स्ट्रैंड. तो ये हो गया N15. इतने तक सबको समझ आया? एवरीबडी अंडरस्टूड टिल नाउ? यस. सो ये है एक बैक्टीरियल सेल 2 हो गया ना. अभी 2 से 4 होना है, 20 मिनट्स में 2 से 4 होगा. तो अगर सेमी कंजर्वेटिव मोड ऑफ रेप्लिकेशन में ये स्ट्रैंड ऐसे स्प्लिट होंगे, एक यहाँ आ जाएगा और वाइट वाला इधर आ जाएगा, है ना. टिल रेप्लिकेशन नहीं, ऐसे स्प्लिट होके वन स्ट्रैंड विल बी गोइंग टू वन सेल, अनदर वन स्ट्रैंड विल बी गोइंग टू अनदर सेल. सो इसमें भी वही है, एक स्ट्रैंड जो ये है यहाँ आ जाएगा और दूसरा ये N15 का है ना, वो यहाँ आ जाएगा. हम तो नॉर्मल आइसोटोप 14 में, नॉट दिस वन, N14 में इंक्यूबेट कर रहे हैं ना, N14 में, तो वाइट ही है. यहाँ भी वाइट हो जाएगा, यहाँ भी वाइट हो जाएगा, यहाँ भी वाइट हो जाएगा, यहाँ भी वाइट हो जाएगा. समझ आ रहा है ना? इतना समझ आया? तो टिल हियर 2 बैक्टीरियल सेल, ये और ये, बैक्टीरिया 1 और बैक्टीरिया 4 इज हैविंग द हेवी आइसोटोप. बीच का बैक्टीरिया इज हैविंग द नॉर्मल आइसोटोप. अंडरस्टैंडिंग? अभी 4 से 8 होंगे ना अनदर 20 मिनट्स में. अभी फिर से इसको स्प्लिट करो. जब स्प्लिट करोगे तो एक वाला जो N15 वाला स्ट्रैंड है ना, सॉरी, N15 वाला स्ट्रैंड है ना, ये दोनों फिर से ऐसे स्प्लिट होके यहाँ आ जाएंगे. अंडरस्टैंडिंग? ये वाला जो N14 है ना, तो यहाँ आ जाए, इसका इधर आ जाएगा. अंडरस्टैंडिंग? और ये दोनों का भी 1 2 3 4 सबका स्प्लिट हुआ. देन रेप्लिकेशन टेक्स प्लेस, राइट? तो उसके साथ क्या होगा? इट विल बी 1 2 3 4 5 6 7 8. तो इसमें N15 है, इसमें N15 है. द बैक्टीरिया, ओनली 2 बैक्टीरिया हैज़ N15. ये N15 कहाँ से आया? जो पेरेंट बैक्टीरिया जस्ट 1 था ना पहले, उसका सेमी कंजर्वेटिव होके उसका DNA यहाँ आ गया है. उसका 2 स्ट्रैंड था ना, उसमें एक स्ट्रैंड यहाँ एक बैक्टीरिया में आ गया है, दूसरा स्ट्रैंड दूसरा बैक्टीरिया में इधर बैक्टीरिया 8 वाला आ गया है. बैक्टीरिया फर्स्ट एंड बैक्टीरिया 8 इज हैविंग द दैट पर्टिकुलर heavy isotope DNA. Understanding? All other, see for example, 2nd, 3rd, 4th, 5th, 6th, 7th, यानी the total of 6 bacteria has normal normal N14, okay, normal, that is N14, and the 2 bacteria is having N15, that is heavy isotope. अगर तुम इंटू करोगे 10 में ओके, यहाँ 1 हो गया तो 10, इफ इट इज 10 देन दिस आर 20 विल बी हैविंग दिस टाइप. 20 से 40 करो, 40 में 20 विल बी हैविंग नॉर्मल एंड अनदर 20 विल बी हैविंग हेवी आइसोटोप. मल्टीप्लाई करो and like 80 into 8 into 10, 80. In 80, that again 20 will be having, or these 20 only, these 20 will be having the heavy isotope and these 60 will be having the normal N14. तो आंसर क्या हुआ? आंसर इज 60. 60 cell will be having the normal N14. 20 will be having heavy isotope. Understood? Yes. The term nuclein for genetic material was used by, it was used by,
Frank.
no, Frederick Mischer, isn't it? It was used by Frederick Mischer, not Franklin, Mischer. The experimental proof for semi-conservative replication of DNA was first shown in, it was first shown in,
Bacteria.
yes, that is bacterium. The same thing about the heavy isotope experiment. Select the correct match. Ribozyme is a nucleic acid. Yes, we got it. F2 recessive parent we can find in the monohybrid cross as well. TH Morgan is for chromosomal theory of inheritance. Mendel giving laws of inheritance. So, which is A is the correct match, all three are wrong match. The final proof for DNA as genetic material came from the experiments of, many times this question has been repeated, Hershey-Hinchey's experiment. The molecule that can act as genetic material must fulfill the traits given below, except, which you accept in a sense you need to find the non-correct one. It should be unstable. Option A. Yes, it should be unstable structurally and chemically. No, it should be stable. It should be stable. We got the answer A. A is the correct answer here. Correct option. It has got the wrong statement. The Taylor conducted the experiments to prove semi-conservative mode of chromosome replication. Option D. Faba beans. Yes. Which of the following rRNA acts as structural RNA as well as ribozyme in bacteria?
23 sRNA.
Yes, 23 sRNA. Option A is the correct answer. In sea urchin DNA, which is double stranded, 17% of the bases were shown to be cytosine. 17%, which means another 17% is guanine. And the percentage of the other 3 bases, tell me.
Option A.
Yes, so this should be 33 and this should be, oh wait, This should be 33. Option A, 17%, 33, 33. Option A is the correct answer. Which one of the following is not applicable to RNA? 5' phosphoryl and 3' hydroxylase, heterocyclic nitrogen bases, Chargaff's rule, complementary base pairing. Can we apply Chargaff's rule for RNA? Chargaff's rule can be applied for anything which is double stranded. Here RNA is single stranded. So we cannot apply Chargaff's rule for RNA. Transformation was discovered by Griffith experiment, transformation principle, he gave it, transformation principle. Which of the following statement is correct regarding the process of replication in E. coli? The DNA dependent DNA polymerase catalyze polymerization in 5' to 3' as well as 3' to 5' direction. The DNA dependent DNA polymerase catalyze the polymerization in 5' to 3' direction. The DNA dependent DNA polymerase catalyze polymerization in one direction, that is 3' to 5'. The DNA dependent RNA polymerase catalyze the polymerization in one direction, that is 5' to 3'. Tell me. In which direction the polymerization takes place? DNA polymerase 3, especially in the E. coli, that is prokaryote, does 3' to 5' polymerization takes place? No. And DNA dependent RNA polymerase, why? We are talking about the process of replication, why RNA polymerase comes. So this is also wrong. It is DNA dependent DNA polymerase catalyze the polymerization in 5' to 3' direction. Always the DNA polymerase catalyze the polymerization in 5' to 3' and 3' to 5' is always exonuclease activity, exonuclease activity, proofreading, many things happens 3' to 5', but 5' to 3' is always polymerization, whether be it in eukaryotes or maybe in prokaryotes, always the polymerization takes place in 5' to 3' direction only. Understanding? Even if you like having confusion in this particular question, you can answer by the elimination technique. It cannot happen 3' to 5', 3' to, so these two are eliminated. And here it is given RNA polymerase, this is also eliminated. Okay. Sometimes when you stuck with the answer, if you don't know correctly what the correct answer is, try reading the question, try eliminating the other options. When you could able to eliminate two options, then it will become like, if you could eliminate about this A and C because of 3' to 5' direction is written, for example C. So you got this and this as a, if you have the confusion between these ends, there is still 50% chance of you getting the correct answer, isn't it? And even if you see like very closely read 2 times, you can definitely able to eliminate this option as well, because logically DNA dependent RNA polymerase क्यों आएगा DNA replication में? DNA dependent RNA polymerase comes in transcription only. RNA polymerase का यहाँ कोई काम नहीं है, तो eliminate it. Left is B, so B should be the correct answer. Statement 1, DNA polymerase catalyze polymerization only in one direction, that is 5' to 3'. Tell me. अभी तो बताया था. DNA polymerase catalyze the polymerization only in one direction, that is 5' to 3'. During replication of DNA, on one strand the replication is continuous while on the other strand it is discontinuous? हाँ, जो स्ट्रैंड में कंटिन्यूस है वो लीडिंग स्ट्रैंड है, जिसमें डिसकंटिन्यूस है वो लैगिंग स्ट्रैंड है. Isn't it both the statement true? Both 1 and 2 are correct. Yes. During DNA replication, Okazaki fragments are used to elongate. Elongate करते हैं लैगिंग स्ट्रैंड को, तो एलिमिनेट कर दो लीडिंग, एलिमिनेट कर दो लीडिंग को, में बचा लैगिंग. द लैगिंग स्ट्रैंड टूवर्ड्स द रेप्लिकेशन फोक, द लैगिंग स्ट्रैंड अवे फ्रॉम द रेप्लिकेशन फोक. देखो ये रेप्लिकेशन फोर्क है, सो इफ इट इज 3 प्राइम टू 5 प्राइम, 3 प्राइम टू 5 प्राइम, सो DNA replication taking place from 5 prime to 3 prime. So okay, 5 prime to 3 prime. So यहाँ इस तरफ जा रहा है, ये 5 prime to 3 prime, इस तरफ 5 prime to 3 prime, इस तरफ. So it is, ये replication fork है, तो the lagging strand is going away from the, isn't it? इन अ सेंस ये पहले तैयार हुआ फिर ये बाद में तैयार हुआ, लेकिन इट इज़ गोइंग अवे फ्रॉम द रेप्लिकेशन फोर्क, इस तरफ नहीं जा रहा. अगर इस तरफ ऐसे गया होता तो इट वुड बी, द रेप्लिकेशन वुड हैव टेकन प्लेस फ्रॉम 5 प्राइम टू 3 प्राइम, लेकिन 5 प्राइम टू 3 प्राइम, 3 प्राइम टू 5 प्राइम, 3 प्राइम टू 5 प्राइम, 3 प्राइम टू 5 प्राइम, तो रेप्लिकेशन इट डजंट टेक्स प्लेस एट ऑल, isn't it? So it is, what is the answer?
Option D.
The lagging strand away from the replication fork. So these particular things are called Okazaki fragment. Okazaki fragments. Select the correct pair. Direction of RNA synthesis. Direction of RNA synthesis is definitely अगर ये वाला this thing, if you see in the textbook ऐसा है ना, this is the RNA polymerase here, so it is always 5 prime to 3 prime. The mRNA is always 5 prime to 3 prime in direction. So the direction of RNA synthesis is from 5 prime to 3 prime. So इसको हटा दो, it is 5 prime to 3 prime. Direction of reading of the template DNA strand, अगर ये 5 prime to 3 prime है तो it would be definitely 3 prime to 5 prime ही होगा. Reading complementary होना चाहिए ना, so it will be, if it is 5 prime to 3 prime, it will be definitely 3 prime to 5 prime is a template strand. Understanding? So correct option हुआ B for Bombay. Any doubt in this? I can explain it to you. No doubt? No doubt? Okay. Yes. So B for Bombay is the correct answer. Given below are two statements regarding RNA polymerase in prokaryotes. Okay. Statement 1. In prokaryotes, RNA polymerase is capable of catalyzing the process of elongation during the transcription. Is it true?
Yes.
RNA polymerase associate transiently with Rho factor to initiate the transcription.
No.
Yes, this is Rho factor. It is actually the sigma factor for the initiation, Rho factor for the termination. So which makes this statement as wrong. Statement 1 is true, but statement 2 is false. A is the correct answer. The given below are two statements. Statement 1, in eukaryotes, there are 3 RNA polymerases in the nucleus in addition to RNA polymerase found in the organelles. Isn't it? RNA polymerase 1, 2, 3.
Yes.
All the 3 RNA polymerases in eukaryotic nucleus have different roles.
Yes.
Yes. rRNA, RNA polymerase 1 will be coding for rRNA, 2 for mRNA, 3 for especially tRNA. So, they have different roles definitely. So, both the statements are true. C is the correct answer. The transcription unit in DNA is defined primarily by the 3 regions in DNA, and these are with respect to upstream and downstream end. That is, the first one is, if this is structural gene,
Promoter structure gene.
yes. So, if this is, so this is 3 prime to 5 prime, yes. So, here in the upstream it is promoter, this is structural gene, and in the downstream end it is terminator. So, B is the correct answer, promoter, structural gene and terminator. Which one of the correct product of DNA dependent RNA polymerase to the given template? This is the template, match it. Which is the correct product of, which is the correct mRNA sequence? This is a template strand, template strand. Which one? T will be coding for A will be coding for complementary, it will be U. So, no, and C, G, T, U,
Option
U. Yes, tell me. Option
C.
Yes, if you see here, it is G, G, it should be U, wrong. G, G, U correct. G, G, U correct. And okay, what is wrong in this? We will find out. Okay, G, G, C, it is given A, which means this is also wrong, which is C is the correct answer. It takes a little time, but fine. We know how to solve this question. That is more important. So, C is the correct answer. Match list 1 and 2. RNA polymerase 3 for snRNA and tRNA. Termination of transcription,
Rho factor.
rho. Splicing of exons, snRNPs. Data box is present in the promoter site. So, D2 A4. Okay, with A4 only we got the answer, that is B for Bombay is the correct answer. What is the role of RNA polymerase 3 in the process of transcription in eukaryotes? RNA polymerase 3, tRNA, 5 srRNA and snRNA transcription. RNA polymerase 3 has this role, transcription of tRNA, 5 srRNA and snRNA. This is 2, RNA polymerase 2, 1, only snRNA not. So, they have asked polymerase 3, which is C is the correct answer. There is a table given in the NCERT, go through that, okay, especially based on that particular question. So, no, based on that particular diagram, that was a question. which one of the following is the sequence on corresponding coding strand if the sequence of mRNA formed is as follows. We need to find the coding strand, okay? We need to find the coding strand. Just tell me which one. Take your time, आराम से, and solve this question. We need to find, again I am repeating, we need to find the coding strand, not the template strand. We need to find the coding strand. This is mRNA. The coding strand is 5 prime to 3 prime. So you can eliminate these two. 3 prime, these two are eliminated just looking at the orientation. Just looking at the orientation, we can eliminate these two because the coding strand is always 5 prime to 3 prime, and the template strand is 3 prime to 5 prime because the mRNA is 5 prime to 3 prime polymerization. So this got deleted. Now tell me with 5, this one 5 prime, B and D. Easy, because see, the mRNA is like same to same as a coding strand, same to same as a coding strand. If you see A है, इधर तो U है ना, U कैसे आएगा? So definitely wrong. By looking at it, we can say it is wrong. A, T, because in place of U, अगर coding strand को पता लगाना है, in place of U T लगा दो बस, rest everything is same. यहाँ T लगा दो, T लगा दो, T लगा दो, यहाँ भी T लगा दो, T लगा दो. U नहीं होना चाहिए बस. Easy. You don't have to, now don't see when you get the question like this, coding strand मतलब mRNA is very same to same as gene. Coding strand मतलब gene है, तो same to same as gene. Okay. Same to same as the base pairs of that particular coding strand. फर्क इतना है कि U के जगह T होगा क्योंकि DNA में U नहीं होता. Don't convert this. See, अगर coding strand पता लगाना है, don't sit there and do it into the template strand. Template strand से फिर से मतलब complementary में coding strand मतलब mRNA दिया हुआ है ना, तुम ये कर मत बैठना कि पहले उसका template निकालूं, template and उसका complementary coding strand निकालूं. ये बिल्कुल मत करना. Why are you wasting time? ऐसे बिल्कुल मत करना, ऐसे बिल्कुल मत करना. I'll tell you why. अगर ये DNA है, okay, this is 5 prime to 3 prime, 3 prime to 5 prime, ये DNA है, okay. So हमें चाहिए mRNA अभी सिंथेसिस हुआ है. mRNA होगा 5 prime to 3 prime, mRNA होगा 5 prime to 3 prime. मतलब ये वाला स्ट्रैंड है ना, दिस इज कॉल्ड टेम्पलेट, अदरवाइज इट इज कॉल्ड नॉन कोडिंग. ब्लू में लिखना था ना, रुको. ओके, फॉर सम रीजन मैं इसको भी हटा दूंगी. वेट, okay, this particular thing is a structural gene, दोनों मिला के. This particular strand we call it as coding strand, otherwise we call it as sense strand. Okay, this one we call it as non-coding strand, otherwise we call it as antisense strand, otherwise we call it as template strand. टेम्पलेट क्यों बोलते हैं? क्योंकि दिस इज द वन विच इज गिविंग द टेम्पलेट फॉर द RNA. तो RNA होगा this is the RNA produced, okay, so it is 5 prime to 3 prime. अगर RNA 5 prime to 3 prime है तो this non-coding strand or the template strand should be 3 prime to 5 prime, which means the coding strand is definitely 5 prime to 3 prime. ये जो भी है ना ABCD, अभी वो नहीं लूँगी, ABCDEF ऐसे सेम टू सेम यहाँ भी ABCDEF ही होगा. Okay, why not, we'll take the ATGC, AUGC, okay. यहाँ क्या होगा, it will be ATGC, यहाँ क्या होगा, TACG. तो TACG has become AUGC. Okay, this is mRNA. अगर देखोगे if this particular mRNA को देखोगे यहाँ 5 prime to 3 prime as a coding strand, सेम टू सेम है लेकिन T के जगह U है. समझ आ रहा है? सो जो mRNA है सेम टू सेम इज द 5 prime to 3 prime coding strand. कोडिंग स्ट्रैंड यहाँ मास्टर है. बस ये टेम्पलेट 3 prime to 5 prime क्यों ले रहे हैं, क्योंकि वी वांट दैट पर्टिकुलर कोडिंग स्ट्रैंड हियर. दैट इज वाई दिस स्ट्रैंड, दिस इज द मास्टर स्ट्रैंड. ये 5 prime to 3 prime है ना, दैट इज द मास्टर वन. उस सेम टू सेम हमें यहाँ mRNA में चाहिए. दैट्स वाई ये स्ट्रैंड को हम कोडिंग स्ट्रैंड बोल रहे हैं, क्योंकि दैट इज द वन सेम एज द mRNA. इसीलिए उसको सेंस स्ट्रैंड बोल रहे हैं, क्योंकि कुछ सेंस बन रहा है mRNA में, सेम टू सेम है. वाई दिस पर्टिकुलर, तुम सोचे होंगे कि मतलब ये टेम्पलेट कर रहे हैं लेकिन हम इसको नॉन कोडिंग क्यों बोल रहे हैं. नॉन कोडिंग इसलिए क्योंकि ये कॉम्प्लीमेंट्री बेस पर दे रहा है, सेम टू सेम नहीं दे रहा, कॉम्प्लीमेंट्री. अंडरस्टैंडिंग? दैट्स व्हाई इट इज कॉल्ड नॉन कोडिंग और एंटी सेंस. एंटी सेंस का उल्टा एंटी सेंस. वी आर जस्ट यूजिंग दिस 3 prime to 5 prime फॉर द mRNA सिंथेसिस. लेकिन क्या हमें क्या चाहिए, हमें चाहिए ये वाला सीक्वेंस, ATGC का सीक्वेंस यहाँ mRNA में, दैट इज AUGC का. बस तुम ये करो कि U के जगह T लगा दो, बस उतना ही करना है, कुछ और नहीं करना है, टाइम वेस्ट नहीं करना है. कभी भी ये 5 prime mRNA को नॉन कोडिंग में ट्रांसलेट करके फिर कोडिंग ऐसे मत करना है. समझ आ रहा है? Okay, it took like 5 minutes for explaining, but you need to remember this. Understanding? Yes. So which is the correct answer? D4 Delhi is the correct answer. हम तो पहले ही ये दोनों, ये हो गया था, ये भी गलत, ये भी गलत. So the correct answer is D4 Delhi. Understanding? Save your time. मतलब within 2 seconds you can do this. बता रही हूँ. Given below are 2 statements. Statement 1, the process of copying genetic information from one strand of the DNA into RNA is termed as transcription. Is it correct? Yes. The transcription unit in DNA is defined primarily by the 3 regions in the DNA, that is promoter, the structural gene and the terminator.
Yes.
So both are correct, isn't it?
Yes, ma'am.
Yes. I want everybody to participate, okay? Everybody participate. In which of, what is the role of RNA polymerase 3 in the process of transcription? Again tell me.
Option A.
हाँ, remember it as RNA polymerase 1 है ना, RNA, rRNA. 1 is always RNA means rRNA. 2 middle, 2 is in the middle, which means mRNA, M for middle. 3 THRE3, 3 for T, tRNA. ऐसे remember. Okay, option A, transcript tRNA, 5S rRNA and snRNA. Understanding? 3. So this is a again repeated question. Identify the correct statement. RNA polymerase bind with Rho factor to terminate the process of transcription in bacteria. We need to find the correct answer. So got it? The coding strand in transcription unit is copied to an mRNA. The split gene arrangement is characteristic of prokaryote. In capping, tell me why B, C and D are wrong. Why? We got the answer first. That is true. In RNA polymerase bind with the Rho factor to terminate the process of transcription in bacteria. The coding strand in the transcription unit is copied to an mRNA. It is non-coding strand. Complementary दे रहा है तो non-coding strand ही दे रहा है ना. It is not the coding strand. Split gene arrangement is the characteristics of eukaryotes. Exon and intron होता है. In capping, methyl guanosine triphosphate is added to the 3' end of hnRNA. No, it is actually 5' end. तो ये भी wrong है. Understanding? A is the correct answer. We need to find the correct answer. With first itself we got the correct answer. Yes. First statement. Which is the one enzyme that has capability to catalyze initiation, elongation and termination in the process of transcription in prokaryotes? That is ट्रांसक्रिप्शन प्रोसेस में इट इज डीएनए डिपेंडेंट आरएनए पॉलीमरेज है, ये नहीं है, नॉट डीएनए, नॉट डीएनए लाइगेज. दिस इज द वन विच हैज ऑल टाइप ऑफ एक्टिविटी. ओके, इनिशिएशन, एलोंगेशन एंड टर्मिनेशन. इनिशिएशन के लिए थोड़ा फैक्टर चाहिए, दैट इज सिग्मा फैक्टर, एंड फॉर टर्मिनेशन इट इज द रो फैक्टर. बट आरएनए पॉलीमरेज इज द वन हु इज डूइंग ऑल दिस, लाइक ओनली एंजाइम. यूकैरियोटिक जैसे आरएनए पॉलीमरेज 1, 2, 3 ऐसे कुछ नहीं है. इट इज आरएनए पॉलीमरेज, इट इज डूइंग द थिंग दैट इज DNA dependent RNA polymerase. Then name the enzyme that facilitates opening of DNA helix during the transcription.
DNA helix.
Name the enzyme that ये बता दो. Again read the question carefully. Read the question carefully. हम रेप्लिकेशन के बारे में बात नहीं कर रहे हैं, हम बात कर रहे हैं ट्रांसक्रिप्शन के बारे में. डीएनए हेलिकेज, डीएनए पॉलीमरेज, डीएनए लाइगेज क्यों ही आएगा ट्रांसक्रिप्शन में, बताओ. ओपनिंग ऑफ द हेलिक्स ऑफ द डीएनए हेलिक्स ड्यूरिंग द रेप्लिकेशन इज डन बाय डीएनए हेलिकेज. लेकिन when it comes to all in one enzyme, यहाँ all in one enzyme है ना, कहाँ गया वो? ओहो, what is the role of, sorry, the only enzyme that has a capability, okay, that is RNA polymerase. The name the enzyme that facilitated the opening of DNA helix during the transcription. Transcription में all in one enzyme, that is RNA polymerase. कोई DNA हेलिक्स, हेलिकेज, DNA लाइगेज क्यों ही आएगा, समझ आ रहा है? सो यू नीड टू लिसन टू, सॉरी, रीड द क्वेश्चंस वेरी केयरफुली. यहाँ ट्रांसक्रिप्शन दिया हुआ है, रेप्लिकेशन नहीं. अगर रेप्लिकेशन होता तो DNA हेलिकेज होता. अभी ट्रांसक्रिप्शन दिया है तो इट इज DNA polymerase is the one who is opening the helix. Understanding? Easy है. Why? Many of the questions, like hardly 10 days, not even 10 days left for the exam. like revise करो, okay? प्रैक्टिस करो क्वेश्चंस को, रिवाइज करो. यू नीड टू प्रैक्टिस. सारे PYQs लगा दो, DBPs लगा दो. नॉट नाउ बट रिवाइज इट, रिवाइज एवरी डे. स्पेशली मॉलिक्यूलर बेस ऑफ इनहेरिटेंस में 14% वेटेज है यार. अराउंड अप टू like 12 to 14% weightage is there for a molecular base of inheritance. So many questions. Around, if you can expect around 10 to 11 questions from this particular, or 12 questions in this particular chapter only 10 to 12, which means has had such a high weightage chapter. You need to revise more. It is a big chapter also. It takes time, but revise. अभी DPPs मत लगा के पढ़ना. PYQs you do it and revise it. Okay. Is sequence from the coding strand of a gene. What will be the corresponding sequence of the transcribed mRNA? कोडिंग स्ट्रैंड दिया है, ट्रांसक्राइब्ड mRNA सेम टू सेम होगा, टी के जगह यू होगा, बता दो. तो इसको हटा दो. सो GA हुई, इसको भी हटा दो. यस, इट इज ऑप्शन ए. कोडिंग स्ट्रैंड, दैट इज द सीक्वेंस ऑफ कोडिंग स्ट्रैंड. mRNA सेम टू सेम होगा, इट विल बी टी के जगह यू होगा, बस. Spliceosomes are not found in the cells of prokaryote.
Bacteria.
D for Delhi is the correct answer, bacteria. Which of the following RNAs should be most abundant in animal cell? The most abundant are rRNAs. Most abundant are rRNAs, which are ribosomal RNA. Ribosomes are more, which means most abundant. Around 80% of all RNAs are rRNAs. Understanding? Then comes tRNA, then comes mRNA. A DNA dependent RNA polymerase catalyzes transcription on one strand of the DNA, which is called कंफ्यूज हो गए हैं इन दोनों में. देखो, DNA dependent RNA polymer catalyzes the transcription on one strand of the DNA, that is अगर ये mRNA है 5 prime to 3 prime, okay, so there will be a bubble here taking place, isn't it? Transcription bubble होगा, transcription bubble ऐसे. Okay, so it is, if this is a RNA polymerase, this is RNA polymerase, catalyze the transcription on one strand. कौन सा strand है ये वाला 3 prime to 5 prime? This is the non-coding, otherwise it is called as template. ये वाला जो है ये कोडिंग है, कोडिंग मास्टर है, वो तो बैठ के सिर्फ देखेगा, वो कुछ नहीं करने वाला ना. सो जो भी कर रहा है ये नॉन कोडिंग टेम्पलेट स्टैंड कर रहा है. नाउ टेल मी द आंसर. टेम्पलेट स्टैंड. ये सी और डी में कंफ्यूज हो गए क्या? वाई व्हाट वाज द कंफ्यूजन? टेल मी. इजी क्वेश्चन है बच्चों, ये बहुत इजी क्वेश्चन है ये. सी इज द करेक्ट आंसर. टेम्पलेट स्ट्रैंड, one strand of the DNA, the DNA RNA catalyze the transcription on this particular strand, that is ये वाला strand. This is DNA polymerase, sorry RNA polymerase. This particular strand is a template strand. In which disorder change of single base pair in the gene for beta globin chain result in the change of glutamic acid to valine? Sickle cell anemia. बहुत easy है. Before Bombay is the correct answer. Select the correct statement about sickle cell anemia. You need to find the correct statement. There is a change in the gene for beta globulin. True. In beta globin, there is valine in the place of lysine. It is valine in place of glutamic acid. It is an example of point mutation, true. In the normal gene, U is replaced by, इधर ये U is replaced by, क्वेश्चन में A है, इधर चला गया है. Okay, U is replaced by A. Is it correct? No, actually the thing is A is replaced by U. A is replaced by U. mRNA में A is replaced by U. Understanding? So this is also wrong. D is also wrong. So जहाँ भी D है,
Option D.
जहाँ भी D है वो wrong. But option D is the correct. Yes. The silent feature of genetic code are, the code is palindromic. No. UGA act as initiator codon. No, it is AUG.
AUG.
Yes. Very good. The code is unambiguous and specific. True. The code, yes. The code is nearly universal. Yes. इसीलिए हम ऐसे इंसुलिन ये वगैरह वगैरह बना सकते हैं ना, because the code is universal. Okay, now choose the correct option. That is C and D are the correct. Yes. C and D are the correct option. D for Delhi is the correct answer. C and D. Against the codon 5' to 3', that is UAC, what would be the sequence of anticodon on the tRNA? mRNA का कोड है from 5' to 3' में. Against the codon UAC codon है. Okay, this is mRNA का UAC. What would be the sequence of anticodon of tRNA? tRNA जब आएगा तो ये tRNA है, तो अगर आएगा तो what is the code? It will be U के जगह complementary, ये A के जगह U, C के जगह G होगा. Understanding? Which means अगर ये 5' to 3' है, now tell me. So this is 5' to 3'. Tell me the answer. Which option? Option?
A.
Option A, that is GUA. 5' to 3', यहाँ से GUA. Very good. This was such a good question and very good. You got the answer correctly. Okay. In a hurry, don't mark it as B. Understanding? Because directly we'll get anyway AUG out of it. UAC से AUG मिल जाता है. Without thinking about the prime here, the orientation, sometimes in a hurry you tend to mark B as a answer, but don't mark B as answer. It is GUA. इस तरफ से read कर रहा है 5' to 3'. Because अगर ये 5' to 3' है, then it would be 3' to 5' here. Understanding? GUA 5' to 3'. GUA. This is the correct answer. Okay? Yes. Statement 1. The codon AUG codes for methionine and phenylalanine. Now AUG codes for only one thing, that is methionine. So ye wrong. This one is wrong. AAA and AAG both codes for the amino acid lysine. This is correct. AAA and AAG codes for lysine. So 1 is wrong, 2 is correct. C is the correct answer. Under which of the following condition will there be no change in the reading frame of the following mRNA? Read the condition A, B, C, D. We need, like, there should be, the reading frame should be same. Okay, there should be no frame shift. No frame shift. Reading frame should be same. When there will be no frame shift, when the codons will be deleted in the multiples of 3. three's multiples. In three's multiples. For example, A3, this 3 code for 1, this 3 code for 1, this 3 code for 1, this 3 code for 1 amino acid. Okay, for this 1 amino acid B, C, D, E amino acid को कोड करता है, मान लो. इंसर्शन ऑफ G एट फिफ्थ पोजिशन 1 2 3 4 5. यहाँ G को इंसर्ट करेंगे तो फ्रेम शिफ्ट हो गया. फ्रेम अगर शिफ्ट हो गया तो रीडिंग it will be changed in the reading frame. Okay, so which means this is wrong. Deletion of G, यहाँ से G को हटा दिया तो then again frame will be shifted. So this is also wrong. Insertion of A and G at fourth and fifth, this is not in the multiples of 3. अगर deletion of GUG इधर sorry GGU ये पूरा हटा दिया, इसको पूरा हटा दिया तो अमाइनो एसिड क्या बनेगा, A B C को पूरा हटा दिया D E, लेकिन रीडिंग फ्रेम तो चेंज नहीं हुआ ना. नेक्स्ट अगर जाएगा तो F G H I, इट विल बी जस्ट C को डिलीट कर दिया, रीडिंग फ्रेम को कुछ फर्क नहीं पड़ा है. व्हेन इट डिलीटेड और इंसर्टेड इन मल्टीपल्स ऑफ 3, द रीडिंग फ्रेम कुछ नहीं होता. अंडरस्टैंडिंग? यस. सो दिस वन, डिलीशन ऑफ GGU फ्रॉम 7th 8th and 9th पोजिशन. हाँ, मतलब कहीं कहीं और डिलीट कर दिया, एडिशन कर दिया तो रीडिंग फ्रेम शिफ्ट होने की चांसेस है, बट इट इज़ लाइक 3 3 3, यहाँ मल्टीपल्स ऑफ 3 में ही डिलीट कर दिया. अंडरस्टैंडिंग? इफ़ यू डिलीट दीज 3, लाइक इफ़ यू डिलीट 6 7 8, इफ़ यू डिलीट 6 7 8, देन ऑल्सो फ्रेम में शिफ्ट हो जाता है ना, फ्रेम में शिफ्ट हो जाता है. या इफ़ यू डिलीट लाइक 8 9 10, अगर मान लो कि तुमने ये सबको डिलीट कर दिया तो अगेन शिफ्ट हो जाता है. तो इट शुड बी लाइक यहाँ 3 यहाँ 3, व्हाट बेस पे इट कोड्स वहाँ डिलीट कर दिया तो देयर विल बी नो फ्रेम शिफ्ट. अंडरस्टैंडिंग? सो इट इज़ 678. Which of the following feature of genetic code does allow bacteria to produce human insulin by recombinant DNA technology? Easy question this one is. Option C, that is the genetic code is nearly universal. If there is 999 base pair in an RNA that codes for protein with 333 amino acid and the base at position 901 is deleted such that the length of RNA now becomes 998 base pairs, how many codons will be altered? Tell me. तो जो भी है, so it was like 999 was there. यहाँ 901, तो which means it was coding around 333. So यहाँ till 900 till 900 divided by 3 कितना? सो इट इज़ 300 बेस पे. यहाँ तक तो ठीक है. 300 अमाइनो एसिड्स, 300 अमाइनो एसिड्स को कोई चेंज नहीं है. यहाँ 901 है ना, ये डिलीट कर दिया है. फॉर एग्जांपल जो भी हो दैट इज डिलीटेड. सो द फ्रेम ऑफ दीज आर गेटिंग अफेक्टेड. यहाँ से आउट ऑफ 333, 300 और नॉट अफेक्टेड. 900 बेस पर तक एवरीथिंग इज, सब कुछ सही है. 300 और नॉट अफेक्टेड. हाउ मेनी आर अफेक्टेड हियर? Actually there should be 99 base pairs, but one is deleted, now it became 98. Understanding? 99 was coding for how many divided by 3 is, it is 33. 33 base pair को 33 अमाइनो एसिड को कोड कर रहा था, लेकिन अभी 98 ही है. सॉल्व फ्रेम शिफ्ट, यहाँ से फ्रेम शिफ्ट हो गया, फ्रेम शिफ्ट. तो हाउ मेनी अमाइनो एसिड गॉट अफेक्टेड, ऑल्टर्ड? 33. अंडरस्टूड?
Ma'am, can you again explain?
This one?
Yes, ma'am.
So according to the question, see there were 999 base pairs. Okay, तो मान लो कि ये this was the DNA. Okay, double helix. Okay, fine. This was the Oh, RNA. This was the RNA, single. Okay, 999 base pairs were there. Total यहाँ 999 base pairs. 999 is there. Okay, understood?
Yes, ma'am.
Yes. So it codes for the protein with 3. So 999 base pairs, 3 base pair. Sorry यार, base pair कहने के लिए हम sorry. Bases. 3 bases code for 1 amino acid. 3 bases. For example, AUG है. This 3 base code for 1 amino acid, that is methionine. 3 base code for 1 amino acid. Understanding?
Yes, ma'am.
So 999 code for, divide by 3 karo, that is 333 base pair. They have given in the question 333 base pair. So if this is a mRNA, protein में, the polypeptide chain, this one goes on. That is how many amino acid are there? 333 amino acids are there. Okay. Now the condition they have given is yahan like 901 till 900. Okay, this is 900 till 900 bases. Everything is fine. 901 base pair got deleted. 901 base pair nahi, base. I'm really why? Wait. Nine not one base here got deleted. So यहाँ नहीं है. For example, सोच लो कि there was A here and A delete हो गया. Okay. Now this is a condition they have given. Now we need to find how many base pairs, how many codons were altered. Now tell me, till 900 everything is fine, right? Just subtracting it. Till 900 everything was fine. So till 900 bases, how many amino acid it codes? It codes 300. 900 by 3, 300. 300 base pairs सही है ना?
Yes, ma'am.
Oh shit. Wait, sorry. 300 amino acids सही है ना?
Yes, ma'am.
Yes. So now 901's deleted. So which means here 999 minus 900, which is 99 base pairs were here present previously. Now one got deleted. I am drawing the base here. It is now 98. Understanding? You will get the answer anyway, 333 minus 333, but if for explanation purpose, 98. If the first base only got deleted, here it will be frame shift happens. Frame shift mutation. Frame has got shifted. ऐसे. So from the first from the first amino acid only. For example, A was there, A was deleted, A, U, G, C, something was there, and A got deleted. Now it started with UGC. The first amino acid itself is wrong. Understanding? So all the 33 amino acids will be wrong. All the 33 amino acids will be wrong, which is coded by this 98 bases. It was 99 previously. Now it is now one got deleted. It became 98, and all those amino acids in the shifted, which means all the amino acids are wrong. Understanding?
Yes, ma'am.
So it is 33. So this is for the just understanding purpose. With this only you will get the answer. That is 900 till 900 it was correct, which means 300 amino acids were correct, and out of 333 300 are correct, which means 33 is wrong. Understanding?
Yes, ma'am.
C is the correct answer. Which one of the following is starter codon?
UG.
AUG codes for methionine. How many different proteins does the ribosome consist of? Do you know? It is 80 different proteins. Okay, there are the different proteins, 80 different proteins the ribosome consists of. This is a factual question, just remember it, 80 different. Okay.
Okay, ma'am.
Okay, is it time? We have around around 30 questions left, Sakshi. What do we do? It will take around next 30 minutes. It's already time, 4:30. You tell me what to do. Like, I am fine with any, like either.
Ma'am, if you have time, then we will continue.
Okay, fine. We'll continue. The process of translation of mRNA to protein begins as soon as the process of translation of mRNA to protein begins as soon as translation. How it begins, you know, if this is the mRNA 5 prime na capping hai, okay, whatever it is. So if this is the mRNA 5 prime to 3 prime, if this is translating to protein, the first and foremost thing it does is the smaller subunit, the first and foremost is smaller subunit of ribosome comes and attached to particular sequence that we call it as SD sequence, okay, comes and attached in this particular here. Understanding? The first and foremost thing is the smaller subunit coming and attaching on the mRNA. That is the first and foremost thing. Then comes the larger subunit. Okay. If it is in the case of prokaryotes, this is 30S subunits first comes, then comes the 50S. Understanding? This is the first and foremost thing happening in translation. So tRNA activates the larger subunit? No. Smaller subunit of ribosome encounters the mRNA? Yes. The larger subunit of the, no, after smaller only larger comes. Both the subunits join together to bind with the mRNA? No. It is always the smaller comes first and attach to the mRNA, then comes the larger. Always remember.
Yes, ma'am.
Which of the following RNAs is not required for the synthesis of protein? tRNA is required, rRNA, which is ribosomal RNA, is required for the ribosome, mRNA is required, that is the template, and siRNA is not required. Understanding?
Yes, ma'am.
Small interference RNA is not required. C is the correct answer. The first phase of translation is, the first phase, recognition of DNA molecule, amino acylation of tRNA, recognition of anticodon, binding the mRNA to the ribosome.
Binding of mRNA.
First phase of the translation is binding the mRNA to the ribosome. Understanding?
Yes, ma'am.
Wait. The first phase of the, no. So begins with the binding of mRNA to the ribosome, but the phases are A, P and E, right? That is amino acylation, peptidyl and exit of the mRNA. First phase of the translation they have asked the phase, okay? They have asked the phase, that is amino acylation of tRNA is the first phase. This is the beginning of the beginnings, but they have asked the phase, okay? Understanding? Yes.
Yes.
So it is 68. 68 is amino acylation. Amino acylation of tRNA. That is the starting of the first phase of the translation. Binding of mRNA to the ribosome, that is how it begins translation. Okay, but amino acylation starts as a phase. Which of the following is wrongly matched? Operon, structural genes, operator and promoter. That is correct. Transcription, writing information from DNA to tRNA, DNA to mRNA. Very good. 1 second, just give me a. Yes. So it is binding of to the mRNA. It is not tRNA. So which means this is wrongly matched. Translation using information of mRNA, rest everything is fine. Stop enzyme synthesis, okay. So this is about transcription, writing the information from DNA to mRNA. Statement 1, in the lac operon, the Z genes code for beta galactosidase, which is primarily responsible for hydrolysis of lactose into galactose and glucose. Is it correct? Z code for, or Z gene code for beta galactosidase enzyme, beta gal. So, is it true? Yes, Sakshi, quick. Sakshi.
Yes, ma'am.
So, in addition to lactose, glucose or galactose can also induce the lacoperon. No, actually only lactose can induce the lacoperon, not glucose and galactose. So, this is wrong, which means 1 is true, 2 is false.
Option A.
Yes, option A. The lactose present in the growth medium of the bacteria is transported to the cell by the action of growth medium. It has to get in with the action of permease. Permease taken. Permeability, permease give the permeability to the lactose which is in the growth medium to get into the cell. That is permeability. Who gives the permeability? It is the permease who give the permeability. Understanding? A is the correct answer.
Yes.
Match. Frederick Griffith.
Transformation.
Yes. Francis Jacob and Monod.
Genetic.
What? No. It is lacoperon given by Jacob and Monod. And Hargobind Khorana, genetic code. And Messels and Stahl, semi-conservative. Yes, A3. Okay. And B4. So, which is D is the correct answer. Very good. Match list 1 and 2. Gene A in Z and in ZYA, it is Z for the beta gal, I for the this one repressor protein, beta gal, Y for the permease, A for the transacetyl. So, if you see many times, like in here you can see 2 to 3 questions of such thing, okay, such what do you call it as pattern, such pattern has given, they have given in the previous year. Understanding? Very much important. Lacoperon is very much important. Okay, just go through it once, revise it, recall it just before the exam. Okay, so it is, yes, so D1, we got it. C is the correct answer. Name the component that binds to the operator region of an operon and prevents RNA polymerase from transcribing the operon. Who goes and binds in the operon region? It is the
Inducer.
no. See,
Repressor.
yeah, yes, repressor protein. Name the component that binds to the operator region. Should I explain you lacoperon?
Yes, ma'am.
Yes. So the thing is, this is a structural. I'll draw the structural gene. Okay, here. So this is promoter. Sorry. Inhibitor, that is repressor protein, that is again promoter, operator, and this is a Z, Y, and A is a structural gene. Okay, polycistronic. Now the DNA dependent RNA polymerase is transcribing this gene. When it goes here, when it moves forward like this, when it moves forward, it goes and it gives the mRNA, transcribe the mRNA of the repressor protein, the mRNA of, sorry, this one repressor, mRNA of the repressor. Okay, this will be translated to the protein of the repressor. You call it as repressor protein. Understanding? This repressor protein, what happens? It goes here and bind to the operator region like this. It goes and bind to the operator region. Understanding? This protein, which is from this gene, that is inhibitor gene. This is the inhibitor gene. Okay, this will be when the polymerase run through the gene, RNA polymerase run through the gene, it will give rise to the mRNA of the repressor. The mRNA will be translated to the repressor protein. This repressor protein goes and bind to the operator region. Understanding? If this is there, like a barricade here, will the RNA polymerase run through it and make the proteins of ZYA gene? No, right? When this is there as a barricade, it is like stopping the RNA polymerase from transcribing the ZYA gene. Understanding? So this particular thing we call it as switch off. The regulation here, the particular thing is now switch off. There is no proteins of ZYA gene is formed. Why? Why this happens? Because there is no lactose. When there is no lactose in the growth medium surrounding the bacteria, why there will be a gene, that is Z gene, which will be coding for beta galactosidase. What does beta galactosid does? I will just tell you here. Beta gal, I will write beta gal. What it hydrolyze the lactose to glucose plus galactose. Lactose is a disaccharide. Okay, I am really sorry, disaccharide. Lactose is a disaccharide. Glucose and galactose are monosaccharides. So it will be hydrolyzing the lactose to glucose and galactose. Understanding? Who does this? It is beta galactosidase does this, because glucose is a like favorite substrate for bacteria. Understanding this much? So lactose should be there in the medium for this particular bacteria to produce beta galactosidase enzyme to convert to hydrolyze lactose to glucose and galactose. Dekho, if something is present, then it will do the production of that particular enzyme. If nothing is there, like lactose is not there, why simply this bacteria waste the energy on producing the enzyme for the hydrolyzation of lactose? Tell me. Like it's a wasteful process. यहाँ waste हो गया ना, gene regulate होना चाहिए. Gene regulation का मतलब क्या है पता? देखो, we are formed like this as a human being from one like single cell, that is zygote. So each and every cell in my body is having the same type of DNA, right? Same DNA. But what is making my skin cell as a skin cell, eye cell as a eye cell, and the bone cell as a bone cell? Why? They have different properties, they have different functions, right? What do we do with the eye? We see the skin as a barrier. What it does, because of gene regulation, some of the gene in this particular cell of my skin is off. Skin से हम देख नहीं सकते क्योंकि वो प्रोटीन्स जो भी है फ्रॉम इन द आई, वो प्रोटीन यहाँ प्रोड्यूस नहीं हो रहा ना. ऐसे नहीं है कि पूरे बॉडी में सारे सेल सारे टाइप्स ऑफ प्रोटीन प्रोड्यूस कर रहे हैं, ये तो हो ही नहीं सकता. तो टोटली वेस्टफुल प्रोसेस हो गया. तो इन डीएनए, अगर मेरे पास इतना 2.2 मीटर जितना डीएनए है एक सेल में, तो मच ऑफ इट मेरे जीन्स स्विच ऑफ पोजीशन में होते हैं. स्किन सेल के सिर्फ जो भी यहाँ मेलनिन प्रोड्यूस करना हो या वट एवर इट हैज़ टू डू, लाइक जो भी रोल है स्किन का, सिर्फ वो जीन्स ऑन होते हैं, बाकी सारे जीन्स ऑफ होते हैं. समझ आ रहा है तुम्हें?
Yes, ma'am.
तो सेम बैक्टीरिया में भी अगर लैक्टोज प्रेजेंट है तो वो जीन ऑन होगा, लैक्टोज एब्सेंट है तो वो जीन ऑन नहीं होगा. हमारा एवोल्यूशन ऐसे हुआ कि एनर्जी कंज्यूमिंग है, ये एनर्जी कंज्यूमिंग प्रोसेस है, तो इट इज़ लाइक स्विच ऑफ कर दो, अगर नहीं है तो स्विच ऑफ कर दो, वो वाला. ओके, सो नाउ इट इज स्विच्ड ऑफ बिकॉज लैक्टोज इज नॉट देयर. इफ द लैक्टोज इज देयर, आई विल जस्ट हियर आई विल राइट if the lactose is present, okay, present. So what happens here? If this is P, I, P, O and Z, Y, A. So if lactose is present, same, हमारा ये आके this particular RNA dependent DNA dependent RNA polymerase will transcribe this particular inducer gene first, that is repressor mRNA of the repressor. Repressor RNA will be converted into the protein repressor protein. Okay. But if the lactose is present, lactose आके comes and binds here, it acts as an inducer. Lactose, this is the lactose आके bind करता है और बोलता है कि भाई मैं present हूँ इधर, मुझे एंजाइम चाहिए, don't go and bind to the operator. Understanding? Lactose आके ये कहता है कि to the repressor protein that I am present here, I need this particular beta galactosidase and permease enzyme for the rest of the जो भी metabolism. So don't go and bind to the repressor. This lactose comes and bind to the repressor protein. So will the repressor protein goes and bind to the operator now? Will it go?
No.
No. अगर यहाँ free है, the operator region is free, can this RNA polymerase run through this structural gene and make its protein?
Yes.
I mean mRNA then protein. So this will run through and it will transcribe the protein for Z, Y and A gene, and respectively it will be translating this, did they say protein? This is mRNA, I am really sorry, mRNA, and from this it will be producing the protein. Okay. What are these? This is beta gal. What it does, the lactose present है ना, lactose को hydrolyze करता है glucose and galactose. Okay, beta gal. And this one is called permease. This gives the permeability in the cell membrane to easily for the lactose to enter into the cell. Previous question we were doing it. It was a permease who was giving the permeability from in the cell membrane for the lactose to enter into the cell from the growth medium. Okay, this is permeability. It gives a permeability, and this is transacetylase, and it has got nothing not much of a role in this lacoperon. I mean in the hydrolyzation of this particular lactose. Okay, transacetyl, what it does, it transfers the acetyl group from one to another. That is the role of this, and it has got not direct relation with this particular thing. Understanding? So this is about the lacoperon. Now the beta gal is produced. Now the lactose will be hydrolyzed to glucose and galactose, and the glucose is a very favored substrate for the next respiration, whatever it is. So it will go into the next metabolism, that is catabolism. Understanding?
Yes, ma'am.
Yes. So this is lacoperon. So what is this thing? Again read. Name the component that binds to the operator region of an operon and prevents RNA polymerase from transcribing the operon. It is repressor protein that binds to the operator region of an operon and prevents the RNA polymerase from transcribing the this one structural gene. Understanding? This is the repressor protein. Okay.
Yes, ma'am.
So which one of the following act as an inducer? Inducer कौन है?
Lactose.
Very good. Lactose. In lacoperon, Z gene codes for
Beta galactosidase.
yes, beta galactosidase. Match it. Same. I said na previously this pattern question is being repeated so many times. I gene. Repressor. Z. Beta gal. Y. Permease. A. Transacetylase. Which one? D1. So C is the correct answer. Understanding? Yes. In E. coli strain, I gene gets mutated and it is product cannot bind the inducer molecule. Okay, the I gene, which is the repressor gene, got mutated, and its product, which means this repressor protein, I gene ka, okay, this is gene, and repressor protein cannot bind to the, cannot, यहाँ आके lactose को bind करना था ना, but it cannot bind to the inducer, that is lactose को bind नहीं कर पा रहा है because it got mutated. So same mutated mRNA is there, same mutated protein is there. The protein, the result is, the condition they have given is it cannot bind to the lactose inducer. If it cannot bind to the inducer, this repressor protein goes and bind to the operator, right? So it goes and bind to the operator. If it goes and bind to the operator, the whole time, whether the lactose is present or absent, this thing is switch off only. The
Option D.
ZYA will not be translated. Very good, very good. Okay. So you understood, which means
Yes, ma'am.
yes. Now tell me again same question. Try solving it. I
Repressor.
repressor.
Z began.
Yes, beta gal.
A
ट्रांसएसिटाइलेज. Y परमीएज. बता दो कौन सा है ऑप्शन है. D2 and C4. Understood? It is C is the correct answer. Yes. Select the correct match. Alice Jeffries, Streptococcus pneumoniae, Alfred Hershey and Martha TMV, Matthew like Messels and Strand Pisum Setum. This one?
Option D.
Option D is the correct answer. Okay, Jeffrey is it is DNA fingerprinting. Just for your like knowledge, Jeffrey is DNA fingerprinting. Hershey and Chase, bacteriophage. They did the experiment with the bacteriophage, right? And Messels and Stahl,
Radioactive.
That is also they did with the bacteria. This is they did with the bacteriophage, and he did with the bacterium. Yeah, bacteria. And this one, Jacob and Monod is the correct answer. We got it. Okay, so this is the correct answer. D4 Delhi is the correct answer. All of the following are the part of operon except operator, structural gene, enhancer, promoter. हाँ, एनहांसर जैसे कोई कुछ था ही नहीं ना. द इक्विवेलेंट ऑफ स्ट्रक्चरल जीन के इक्विवेलेंट क्या होता है?
Operon.
नहीं, स्ट्रक्चरल जीन माने पॉलीसिस्टोनिक सिस्ट्रॉन्स. सी वन इन यूकैरियोट्स इट इज मोनोसिस्ट्रॉनिक, विच मीन्स एक प्रोमोटर में एक जीन होता है. बट व्हेन इट कम्स टू द प्रोकैरियोट्स, पॉलीसिस्ट्रॉनिक, विच मीन्स एक प्रोमोटर में मल्टीपल सिस्ट्रॉन्स, दैट इज जीन होता है. अंडरस्टैंडिंग? सो विच मीन्स स्ट्रक्चरल जीन माने सिस्ट्रॉन. यस, डी इज द करेक्ट आंसर. यस. Gene regulation governing the lactose operon of E. coli that involves the LAC-I gene product is negative and negative तो है ही, feedback inhibition positive नहीं है. Wait. This two option you can eliminate. Okay. So in this negative and repressible, negative and inducible.
Negative and repressible.
No. See, LAC-I is a repressor. Okay, repressor. This was inducible. ये इंड्यूस, इसको लैक आई के बारे में बात कर रहे हैं, आई जीन के बारे में बात कर रहे हैं. ओके, वी कुड एबल टू रिप्रेस द जीन, बट आई क्या हो रहा था, आई वॉज गेटिंग इंड्यूस ना.
Yes, ma'am.
तो कॉम्प्रहेंशन, इंग्लिश कॉम्प्रहेंशन भी देर विल बी, लाइक हिंदी भी होता है क्या साथ में, if you can choose. So just if you can, like if you know Hindi, you can read it in Hindi also, okay, because comprehension चाहिए, English का comprehension. Okay, whether it is not an inducer, it was a repressor. I was a repressor. I was a repressor. Okay, which was the inducer? Lactose was the inducer. Inducible, it was, this repressor was getting induced, isn't it, but the lactose, which was getting repressible, it was the gene, operator gene was getting repressible. So we could say that D is negative. We need to find about the correct answer for the lac I, that is negative and inducible, because repressor protein prevent transcription. The repressor protein was getting induced by the inducer, that is lactose. समझ आया?
Yes, ma'am. Yes, ma'am.
So D is the correct answer. And this is really good question. ये बहुत अच्छा question था. We need to know about whether it is induced or it's a repressor, but it was getting induced. It was inducible. Inducible in the sense, मुझे रोक सकता था कोई आके. आई एम रिप्रेसर प्रोटीन, मैं आके किसी को रोकता था, लेकिन मुझे भी कोई आके रोक सकता है. दैट इज आई एम गेटिंग इंड्यूस्ड ना.
Yes, ma'am.
Which enzyme will be produced in a cell in which there is a nonsense mutation in LAC-Y gene? Nonsense mutation, in a sense, there are three types. Okay, mutation, one is silent mutation, another one is nonsense, another one is missense. So which gene will be produced in a cell in which there is a nonsense mutation in the LAC-Y gene? See, silent mutation in the sense, वो three bases के बारे में बात कर रहे थे ना, when in terms of, in multiples of 3 base pairs, if there is a mutation, then it will be silent mutation. Frame shift नहीं होता, the reading frame will be same, that is called silent mutation. Nonsense in a sense, for example, there was there was AAA sense. AAA was this thing, base pair, but A mutate होके what happened? UAA became UAA. UAA is a stop codon. It codes for stop codon, right? So it codes for stop codon. UAA, UAG, UGA, 3 codons code for stop codon. तो यहाँ बोल रहे हैं कि एवरीथिंग वॉज गुड टिल द जी जीन, वाई में क्या हो गया, स्टॉप कोडॉन आ गया, विच मीन्स हियर इट विल स्टॉप कोडॉन हो गया. सो mRNA विल बी देयर, बट प्रोटीन सिर्फ इतना ही सिंथेसिस हुआ. जहाँ तक सिंथेसिस हुआ, बीटा गैल ओनली सिंथेसाइज्ड. mRNA was properly synthesized, but due to some mutation here in the Y gene के साथ यहाँ म्यूटेशन हुआ, दैट टू नॉनसेंस, विच मीन्स स्टॉप कोडन आ गया बीच में. सो प्रोटीन सिर्फ इतना ही सिंथेसिस हुआ. समझ आ रहा है तुम्हें?
Yes, ma'am.
हाँ, तो what is a consequence of it? तो यहाँ will there be permease? No. Will there be transacetylase? No, क्योंकि यहाँ स्टॉप हो गया है. हाँ, so only which enzymes will be produced? Beta gal. Only beta gal will be produced. It was really good question. Understood? 3 types होता है ना. Silent mutation, you got it. In frame shift नहीं होता. There will be no reading frame same होता है silent mutation में. Nonsense in a sense, completely nonsense, like stop codon आ गया. And missense in a sense that frame shift हो जाता है missense में. पूरा फ्रेम शिफ्ट ही बिगाड़ दे, मतलब पूरा रीडिंग फ्रेम को ही बिगाड़ देता है, दैट इज मिस सेंस. तो अंडरस्टूड?
Yes, ma'am.
Yes. Now coming to the genome, human genome project, express sequence tags, EST refers to, tell me, easy, from directly from the textbook. Okay, this class is still going on. Class is still going on. Yeah. Option?
C.
All genes that will be expressed as proteins. It is like all genes will be expressed as expressed sequence tags in which the all genes will be expressed as RNA. Okay, this question is repeated several times, just go through the this thing once. So all sequence that are expressed as RNAs, okay, that means express sequence tags. 85B, B for Bombay is the correct answer. The last chromosome sequenced in human genome project was, starting with a small, ended with the very large chromosome, that is
Chromosome number 1.
chromosome number 1. Very good. B for Bombay is the correct answer. DNA polymorphism forms the basis of, it forms the basis for both genetic mapping as well as DNA fingerprinting. This was the main thing. Polymorphism, DNA polymorphism forms the basis for both genetic mapping and DNA fingerprinting. Understanding?
Yes, ma'am.
Yes, for D for Delhi is the correct answer. If a geneticist uses the blind approach for sequencing the whole genome of an organism followed by assignment of the function to different segments, the methodology adopted by him is called. The one thing about the expressed sequence tag, that was a most feasible method we could adapt, and that was the adapted one in the human genome project. Another one we call it as sequence annotation, in which in express sequence tags, only thing which was coding for mRNA or RNA especially, okay, RNA was sequenced. Here in the sequence annotation, the whole genome, regardless of whether it is giving the mRNA or protein, the whole thing was, the whole genome set was, that was approach, another approach was there. That was not that great, so they didn't use it, but they are asking according to the question what was that approach. The another approach was sequence annotation. Understanding? Not the express sequence tags.
Yes.
Yes. DNA fingerprinting involves identifying the differences in some specific regions in DNA are called repetitive DNA. A is the correct answer. Which is the basis of genetic mapping of human genome as well as DNA fingerprinting? That is because of the polymorphism in DNA sequence, that was the basis for genetic mapping and human genome, mapping of human genome, okay, as well as DNA fingerprinting. What is it? It is polymorphism of DNA sequence. It is DNA polymorphism was the basis. Express sequence tags refers to, already we did this, tell me. This one, the genes expressed as RNA are express sequence tags, okay. Which of the following is not required for any of the techniques of DNA fingerprinting available at present? PCR is required, restriction enzymes are required, DNA hybridization is required, but zinc finger analysis, this is not required. Understanding? Anyway, it is 2016 phase 1 question. In which of the following is required as inducer for the expression of lac operon? 5.
Lactose.
Which of the following is required as inducer? Yes, it is.
Lactose.
Oh, it is only lactose. Yes, here it is lactose is used as inducer for expression of lac operon. Satellite DNA is important because satellite DNA. It shows a high degree of polymorphism in population and also in the same degree of the DNA polymerase in an individual, which are inheritable and from the parents to the offspring. This is the correct answer. A is the correct answer. Commonly used vectors for human genome sequencing directly from the NCERT. Those vectors are bacterial artificial chromosome and yeast artificial chromosome. Otherwise, we call it as BAC and YAC. Understanding?
Yes, ma'am.
Last question. An analysis of chromosomal DNA using the Southern hybridization technique does not use. It uses PCR, electroreporting and autoradiography. Here PCR is not used. Whatever the DNA you get, that will be sequenced. The electrophoresis is used, plotting is used, autoradiography is used, but PCR is not used. Understanding? So this was like 2014 question. Okay, fine. You just know what is the answer to this. That is, PCR is not used. PCR used for amplification. Here we don't want amplification in southern blotting. That's it. So that is the end of the PYQs of molecular base of inheritance. See, in last like 10 years, how many questions have come. So it is very much important for you to go through the lesson, recall it, revise it. Understanding?